Good afternoon. My name is Mike Shelley, and I have the pleasure of chairing this session, which is an invited talk by Itai Cohen. So for those of you who do not know Itai, he is a physics professor at Cornell, and I know him through many other things, many other areas of science aside from what he's going to talk about today, which, as you know, has to do with the flight of the fruit fly. I know him from work that he's done in complex fluids, the dynamics of suspensions, origami materials, and most especially from his fabulous work on the dynamics of mosh pits. And I'm going to turn the floor over to Itai now, and I think you'll have a good time. Let's welcome him, please.
This is Drosophila melanogaster. That's actually me. That's Drosophila melanogaster. Isn't he beautiful? Okay, I sense the skepticism. But this animal really can do some amazing things. And to get us started on why I think that's the case, I'd like to demonstrate a simple controls problem for you. So I'm going to try to balance this rod on the tips of my fingers. And so the way this goes is that this rod is trying to tilt over. It's an inverted pendulum. And my hand has to adjust itself, adjust the position, so that I get under it fast enough. Now, it turns out that this is a control problem that most of you in the audience can do. But once we get to objects that are smaller, say the size of this laser pointer, this problem becomes impossible for humans to do. So it turns out that the time scale for adjustments scales with the size of the object. And so already at 10 centimeters, this is a control problem, and I urge you to try this at home after the talk, that is very difficult to do. Now, Drosophila melanogaster are subject to aerodynamic instabilities whose time scales also are determined by their size. But Drosophila melanogaster are 1 millimeter tall. And that means that they have to make these corrective adjustments 100 times faster than what it would take to balance this laser pointer on the tip of my finger. Pretty incredible. In my lab, we do a lot of work to image insects in high speed and then develop automated algorithms for extracting the wing and body kinematics from these high-speed movies. And these are the kinds of films that we might make. On the three faces of the cube are the three movies that we would take of the front, the other side, and the top of the fly. And then the faithful reconstruction that our algorithms spit out from these movies. And the first thing that I want you to notice is that they are beautiful. These animals are beating at something like 30 times in the blink of an eye. And their wing strokes are these figure-8 masterpieces going back and forth and back and forth. And my students get to look at this every day when they go into the lab. So using these kinds of movies, we can start to think about the various maneuvers that these animals make. And for example, we can try to test some ideas that have been put out there. So if you open up Alexander's Nature's Flyers, you can see that there are ideas about how insects are supposed to produce a forward thrust. And the basic idea here is that insects are supposed to take their stroke plane and tilt it at an angle so that some of the lift forces are redirected in the forward direction. And that's how you generate a forward thrust. And we see this sometimes. When the flies really need to get out of there fast, they'll do this. But oftentimes, they'll do something more akin to this, where they keep their stroke plane nearly horizontal, and yet they're still able to produce a forward thrust. How are they doing this? We can take a look at the difference between a hovering fly and a fast-moving fly. And so in the bottom right here, or left for you, I'm showing you what's known as a ball and stick diagram. The ball corresponds to the leading edge of the wing. The stick corresponds to the cord. And you can see that they're making these beautiful figure-8 stroke patterns. And sure, these look a little bit different, but mostly, actually, those differences are quite subtle. So if we tease this type of stroke apart, into the three components that define a wing stroke, we see differences in all of these three components. So the top graph shows you the stroke amplitude, and so that's the main motion that the wing is making, and it's sinusoidal as the wing swings from the front to the back. The middle one is the deviation angle. It's how much the wing deviates from the horizontal. And you can see that it has two peaks, one in the front of the stroke and one in the rear of the stroke. And that's because the animal lifts its wing in the front and lifts its wing in the back as it's stroking back and forth. And then the last one has to do with the pitch. So it turns out that the pitch of the wing is related to the angle of attack. It's defined as 0 when the wing is horizontal and then goes all the way up to 180. And insects are usually flapping their wings at a 45-degree angle on the way forward and 135 degrees on the way back. And if we go back to the stroke angle, that's what you're seeing in the bottom plot, where it goes forward at 45 and back at 135. Now, all of these are slightly different. The red and the blue curves corresponding to the fast-moving fly and the hovering fly, respectively, are different from one another. But which differences are important? How do we solve that problem? And so to do that, to solve that problem, we turn to simulations. And if we put in the flight kinematics for the fast-moving fly, we're able to get a fly that moves forward quickly. So it's able to produce that thrust. In fact, if we put all the curves from the hovering fly, we can get an animal that's moving at close to 0 velocity. So that's this data over here. And if we take the stroke curves for the fast-moving fly, we produce an animal that's moving at a high relative velocity. But the nice thing about simulations is that we can now mix and match. So for example, I can take the stroke plane from the slow-moving fly, the stroke curve from the slow-moving fly, the deviation curve from the fast-moving fly, and the pitch curve from the slow-moving fly. And you can see that the velocity of the fly hasn't changed one bit. And that basically means that the deviation angle, that's irrelevant for producing forward thrust. On the other hand, if I take the pitch curve from the fast-moving fly, it doesn't matter what the other ones are, I always produce a fast-moving animal. In fact, I can do one better. I can take all of the curves from the hovering fly, and if I just shift the curves for the pitch downwards, I can generate a fast-moving fly, or vice versa. I can take all the curves from the fast-moving fly, shift the pitch upwards, and I generate a hovering fly. What's going on? Well, let's see what this means for the fly here. So the fly is flapping back and forth at 45 on the way forward and 135 on the way back. But if I lower the angle uniformly, if I reduce the pitch angle, then what the fly is doing is slicing through the air on the way forward, and on the way back, it doesn't quite get to 135, and that means that it's smashing on the way back. And so these animals are essentially swimming through the air as their way of producing forward thrust. It's not quite that simple, because as they start moving forward, their body velocity contributes to the wing velocity, and that affects the drag forces acting on the wings. It's the same kind of thing that happens when you jump out of an airplane and you reach terminal velocity. At some point, there's going to be a speed at which the drag from the body velocity compensates for the thrusts that the insect is able to produce. So we can write down a very simple reduced order model of this, and it basically relies on the intuition that we all have when we stick our hand out of the window of a car, that the drag forces are low when the hand is parallel to the direction that we're moving in, and the drag forces increase as we make that hand more perpendicular. And this is what that drag coefficient looks like, and the drag forces are basically scale like the wing speed squared. You can now add in the body velocity into this calculation. So here I'm showing you the drag force on the wing on the way forward, where the speed is now added to the body velocity, and on the way back, where the speed is now subtracted from the body velocity, where we subtract the body velocity from the speed. And if you keep the lowest order terms, the part in the parentheses has to be 0 at all times, and that leads to a very simple law. It says that the velocity that the fly reaches is proportional to the wing speed times the angle of attack difference between the forward and the backward directions. That's it. A very simple law. We can test that in the experiments. And so if I plot for you the flight speed as a function of the paddling angle, what you're seeing is that the data fall on a line, and the prediction, which is the dashed line over here, matches pretty well to the data. Now, these data make me very sad, because there's about 16 movies that we had to analyze. And each movie was something like 30 wing strokes. And each wing stroke was 40 moments in time. And for each moment in time, we had to extract 3 angular and 3 center of mass coordinates for each wing, body, and wing. And that means that this plot right here is really 40,000 different data points, all summarizing a linear relationship. And that is why this field is so hard. And that's why making automated extraction of the kinematic data is so important for progressing and figuring out these types of dynamics. We can do other things. For example, if we put in an LED arena, we can induce the fly to make turns. And so in this particular case, we show it an LED movie of the world moving to the right. And then the fly follows that and produces a saccade, which is an in-plane turn to the right. So this is a 140-degree turn to the right that's, again, induced by this LED arena that it's observing. And what's nice about turns, in-plane turns, is that you all now know how this works, because turning is nothing more than moving forward with just one wing. Is it clear? OK, well, lucky for you, I brought a demonstration to help illustrate that point. Thanks to the folks at Georgia Tech, I have here a swivel platform that I'm going to lay on the ground. And then I brought my wings with me. So I even painted them for you. And I'm going to try to get on this thing without killing myself. So there I go. All right. Now, so if I flap my wings symmetrically, nothing much happens. Although, Mike, can you attest that you can feel the wind? You can. OK. But now, if I move forward with the right wing, so slicing through the air, smashing, then I can turn to the left. And if I do it the other way, I can turn to the right. And that's how it goes. Now, notice that I did not take off. And that's because the amount of drag forces that I need in order to produce something like a rotation are minuscule compared to the forces that I need to produce lift, something like 1%. And this really represents the main concept behind why this strategy works so well. So if we look at the curves for lift and drag coefficients as a function of the angle of attack, what you see is that the lift forces have a maximum at 45. That means that if I change the wing pitch angle just a little bit about that maximum, I don't change my lift forces to first order. On the other hand, the drag forces go right through 45. And so by changing the angles a bit, I can change my drag forces a lot, but keep my lift forces sufficient to keep me in the air. And that's why this strategy works. Now, this idea of differential drag has a long and distinguished history. It turns out that the Wright brothers were battling with this. Everybody knows the Wright brothers were not the first people to fly, right? There were many people flying before the Wright brothers. That's not what they became famous for. No, what the Wright brothers became famous for was the first stable flight. What do I mean? Well, flying is not like driving a car. So if I have an airplane, it turns out that you can't just steer it by changing your rudder. Airplanes are subject to roll instabilities. So they tend to roll from side to side. And the strategy that people were using at the time is that when the airplane started to roll to the right, the pilot would move a little bit to the left. And when the airplane started rolling to the left, the pilot would move a little bit to the right. And you can see this was a very bad idea. And lots of people died using this technique, including Lilienthal. No, so what the Wright brothers did, they figured out a way to warp the wings. So you would pull a lever, and then that would increase the angle of attack on one side of the airplane. So if you started rolling, you'd increase the angle of attack. But that would also increase the drag, and so the airplane would yaw. So what the Wright brothers had to do was invent a triple axle controller that compensated for this yaw with the rudder. And that's what they got their patent for, and that's what they were so famous for. But I think you'll agree with me that this is kind of a kludge, right? Because this isn't a natural mechanism. This is something where you have to compensate for it. Now, the Wright brothers were amazing engineers. They figured out the shape of propellers to something like 90% of their modern-day efficiencies. So I think they would have been really impressed with the fact that flies have figured out a way to interleave drag and lift forces in a much more elegant fashion. Now, at the controls was Wilbur Wright. What's happening with the insects? How are they controlling their flight? So to try to answer this question, my student at the time, now Professor Ristroff at NYU, concocted the following experiment. He took a brush from the Gordon Brush Company, so there it is. And what he noticed is that if you take a clipping from that brush, you can glue it to the back of the fly. And these clippings happen to be magnetic. So now we put the flies into our apparatus. And in addition to triggering the filming of the flies in our field of view, we're now going to dump a current across a pair of Helmholtz coils. Those Helmholtz coils are going to rotate the fly in midair and cause a midair perturbation. Okay, that's the idea. How did it go? This is the first movie that we ever captured with this. You can see the pin sticking out the back of the fly. And right around here, for one wing stroke, we apply the magnetic field and torque the fly. Okay, I'll show it to you again. Here it comes. And then right around here, for one wing stroke, we put on the magnetic field. And the amazing thing about this movie is that if you take a look at the orientation of the fly before and after, it's identical. These flies have a mechanism for autocorrection. How do they do that? If I look at a pitch perturbation, so this is one where we glue the pin straight out and have the Helmholtz coils top and bottom, the fly ends up changing how far forward the wings are stroking. And so when you pitch forward, the wings go more in the front. That produces a torque that then, from the lift forces, that then rotates them back. And vice versa, if the pitch is towards the back, then they don't flap as far in front, and then the torque from the rear forces torques them forward. And so the question really becomes, if I plot for you the front stroke angle as a function of time, where's that recipe coming from? Those are the angles, but how does the fly know what angles to choose? I'm not going to take you through all of the control theory that we had to do to figure this out, but I want to point out the intuition behind it. And so, for example, if I take now the body pitch velocity, that's this curve right here. And if I now take the integral of that, which is the total angular displacement in the pitch direction, that's this curve right here. And if I add these two together, they go right through the data. So what I'm telling you is that the flies can measure their angular velocity. They then integrate that to get the total angular displacement, so they know calculus. And then they sum the two terms together and pump them into the steering muscles in the wings. And this is the control law that they're governed by, with a Kp, a proportional coefficient for the angular velocity, and a Ki, an integral coefficient for the total angular displacement. And this works for roll and for yaw. In roll, the insects have to flap the wing that's down a larger stroke amplitude than the wing that's on top. And for yaw, they basically do the same thing that I demonstrated for you on the rotating platform. So in all of these cases, it just depends which degrees of freedom you're actuating, but all of these are governed by the same proportional integral controller. proportional to the velocity and the integral of that, which is the angular displacement. And this is the kind of circuit that we like to write down for these. You basically have an external torque that's acting on the fly. That's our magnetic field. The fly measures these torques using vibratory gyroscopes. It then has some time delay, which then gets the signal from these vibratory gyroscopes, after some time delay, gets integrated, and the proportional part all get fed into the steering muscles, which then alter the wing motions, which then produce new torques, and so on. And I know what you're thinking. These are highly contrived experiments. So is this really biologically relevant? And to help demonstrate biological relevance, I want to show you this video. Stick with me. I know what you're thinking, upstate New York, but just stick with me. Did you see it? Okay, I'm going to play it for you again, but I want you to look really closely at the bottom right of the screen. Because at the bottom right of the screen is a fly. And as the gun goes off, that fly is performing a roll perturbation, just like they do in our experiments. I'm going to play it for you one more time. Bottom right, there it is. Watch as the gun shoots, the fly experiences a roll perturbation, and then does the same corrective wing mechanisms that I talked about in order to respond. So I think the answer is unambiguously yes. At least in upstate New York, these are absolutely biologically relevant experiments. I also told you that the fly has these kinds of control circuits, these vibratory gyroscopes, in a circuit that really resides in the back of the fly. And we know that it resides in the back because of this movie. So here's a fly that's going to take off, and it's going to fly stably. So it doesn't have any problems stabilizing its flight. But can you see what the problem with this movie is? Yeah, these flies have no head. Their head has been clipped off, and their neck cauterized. All of the control circuits that these animals need in order to fly stably are located in the back, in what's known as their ventral nervous cord. The head is overrated. The vibratory gyroscopes that I was talking about are actually remnants of the third and fourth wing of all Diptera. So all Diptera used to have 4 wings. The hind 2 wings became these nodules called halteres, and these beat in antiphase with the wings. And what happens is that when the insect turns, these nodules get left behind. There's a campaniform underneath them, and so as they pull on the campaniform, neural signals are elicited. And the idea is that those neural signals then go into the steering muscles. So here is an X-ray tomography image from the group at Oxford, beautiful experiment, where they show the warm colors are the main drivers of the wing stroke, and the cooler colors are 4 out of the 12 steering muscles that are guiding these very subtle wing motions. Okay? Now, one of these 4 muscles here is called the basilar 1 muscle, the B1 muscle, and it's thought to play an important role in pitch. And so the idea is that nowadays we can genetically engineer flies so that we can chronically silence these motor neurons. And that means we turn off the muscle. So here I'm showing you confocal stacks of the B1 motor neuron highlighted, and using a set of techniques that, if you want, I'm happy to talk to you about later, we can do the following experiment. We can either have the B1 motor neuron and leave it alone, or we can silence it permanently. Just take it out of the game. What happens? So if I have a normal fly, it starts off with some pitch angle, then we apply the perturbation, and it does the corrective maneuver, as I showed you before, using a PI controller. So the green curve is just a PI controller fit to the wing stroke, the forward wing stroke angle. If I do the same thing with a silenced B1 motor neuron, what I find is that, first of all, the flies have a hard time maintaining their initial pitch. And after the perturbation, they're still oscillating, but now about a new angle. So they forgot where they came from. We can fit the front forward angle again, but this time we only need a proportional controller. The integral term has been taken out. You can do this more systematically and show that it's only the integral term that gets modulated. So the integral gain ki is lower than what it is for the controls. The proportional gain and the time delay are exactly the same. So what I'm telling you is if I take out the B1 motor neuron, I take out this term in my PI controller. And the question is this true only for chronic silencing? Maybe this is just some compensatory mechanism that the fly came up with because its neurons are silenced. Well, to help elucidate that, we did a B1 inactivation experiment. So what this means is that the fly is living a normal life until at some point it encounters this green light. And when it encounters that green light, the B1 motor neuron gets silenced. And it loses its ability to maintain its pitch angle. And then when we turn the light off, it regains that ability again. You can look at the PI controller. Again, the integral portion of the controller is gone. Alternatively, we can do the opposite. We can activate the B1 motor neuron. So this fly is living a normal life until at some point we turn on a red light, and now it overcompensates, and what you end up getting is a fly doing a double backflip. In all of these experiments, the roll control circuit, the roll PI controller, is unaffected. And so that brings us to this idea that maybe there are separate PI controllers for yaw, pitch, and roll acting in parallel in order to govern these 3 degrees of freedom. Now, is that it? Is it just that simple? No. We know that, for example, the flies prioritize roll. So if the fly is both rolled and pitched, it's going to correct the roll degree of freedom first. We also know that when we do this to the fly, by switching the magnetic field back and forth, we can create a fly that has a quintuple helix with a triple twist, I think the first time ever. And when we turn that magnetic field off, you'll see right around here, after 1, 2, 3, or 4 wing strokes, this fly recovers. Now notice that it didn't turn back 8 times. And that means that the controller can't be purely linear. It has to have a mod function. How do these circuits play out in this ventral nervous cord? How do we get the information from the sensory apparatus, which are these green things here, there we go, to the interneurons that connect the sensory neurons to the motor neurons? And how are they affected by descending neurons that are coming down from the brain, acting to influence this whole thing? So to go after these problems, we grouped with a team at the Howard Hughes Medical Institute, and we developed about 220 genetically modified flies where we can tackle all of these different neurons in the ventral nervous cord. And so the program is that we now go in, we silence, we activate, and try to figure out what each neuron is doing in this complicated but fascinating phenotype. So let me finish by asking the question, is all this machinery necessary? What happens if we don't have halteres? Well, if we take the halteres and we glue them down to the back of the fly, basically, they can't fly anymore. Okay? They just fall out of the air. But you can do a linear stability analysis where you can show that if you were able to somehow slow the rotation time to a sufficient degree, you could get these animals to be passively stable, or at least stable enough that a different sensory modality takes over. And you can do that by taking a dandelion seed, crushing it, and then gluing it to the butt of the fly. And when you do this, even though the halteres are glued down, these animals can fly no problem. We've rescued the ability to control flight. In fact, there are animals that use this technique all the time. This is the woolly aphid, commonly known as the fuzzlebutt. And the fuzzlebutt has all of these beautiful hairs sticking out of it. And these hairs essentially make it passively stable to flight. So why not just use this technique? Why do we need these halteres at all? Well, in the same way that a passenger airline jet is stable but not very maneuverable, These hairs are good for stabilization, but they sacrifice your maneuverability. By trading these hairs for a couple of halt ears, as sported by this neon yellow halt-eared fly, you can gain maneuverability, which makes you a better predator and helps you avoid predation. So there's a real evolutionary pressure to develop these kinds of vibratory gyroscopes and this type of control algorithm for stabilizing your flight. So let me finish by saying that, or by showing you a picture of a fly on top of a reproduction of the patent by the Wright brothers, and say that 350 million years ago, before flies take to the air, before insects can learn how to fly, there are no trees. All plants are less than 3 meters tall. No flowers. So the ability of insects to take to the air and control their flight has a profound effect on the ecology of this planet. This is Drosophila melanogaster. Isn't he beautiful? Thank you. I want to take just a second to recognize the hard work of all the people who worked on this project. The folks in blue here were the ones who were responsible for the aerodynamics, and the folks in red are the ones that we are pursuing all of the neuroscience with. And with that, I'll take any questions that you might have.
Please.
Thanks for a fun talk. 2 quick questions. You got a PI controller. Is there any sign of D? And, okay, 3 questions. Really just one neuron, and can it be fast enough?
One neuron, meaning the B1 motor neuron?
Yes.
Yeah, so it turns out, so let me answer the first question. No, there's no evidence of a derivative part of the controller. The second, and all I can tell you is that a PI is sufficient for stabilizing flight. So that's good enough. Every one of these steering muscles is innervated by one motor neuron. That's what makes this such an attractive system for studying these types of perturbations. There's very little redundancy in the ventral nervous cord.
And the neural processing speed, I guess it is fast enough. That's consistent with...
Neural processing speed is on the order of a millisecond. And so what we see is that the actuation of the correction maneuver happens on the scale of about 5 milliseconds, one of the fastest responses in the animal kingdom. Nevertheless, it means that you only have a few neural synapses before you get to the actual motor, before you get to the muscle.
Neat, thanks.
Okay, thank you very much.
Thank you. Let's thank Itai again, please.